Hi friends, it's Sara. Most antibodies don't fail because the biology is wrong. They fail because they're difficult. They don't express well. They aggregate, they fall apart, or they turn into syrup the moment you try to formulate them. And the frustrating part is that these problems rarely show up alone. They show up together, quietly. And the full picture often doesn't become clear until after a company has already spent millions of dollars on research and development. Researchers at Regeneron asked a simple, dangerous question. What if we could predict an antibody's developability before ever making the first test batch? Imagine taking those millions of dollars and investing them in other molecules, or expanding production capacity to help reduce drug costs. They're not asking this out of curiosity. They're asking out of experience, and with an eye toward what's possible next. So these all-stars at Regeneron put together a program they call PROPERMAB. There is a link to the paper in the description. At a high level, PROPERMAB pulls together information from the antibody sequence and predicted structural features. They then line all of that up with measured antibody developability data and glue it together with a machine learning model. Not to find a perfect antibody, but to find one that's well-behaved, civilized, proper. A proper monoclonal antibody. Yes, that is absolutely what they named it. And honestly, I respect it. So let's get into how they built this thing. Figure 1 in the paper is a great overview of the whole workflow. They start with a collection of antibodies with known sequences and experimentally measured developability properties. First, they run those sequences through what they call a sequence featurizer. You can think of this as a protein language model, kind of like ChatGPT, but trained to recognize amino-acid patterns instead of words. Things like glycosylation motifs, phosphorylation sites, charge patterns. All of that gets encoded into features the model can work with. In parallel, they feed those same sequences into an antibody structure predictor called immune builder. Those predicted structures are then used to identify things like charged patches and hydrophobic regions on the surface of the antibody. Precisely the kinds of features that tend to show up later as aggregation, viscosity, or solubility issues. And this is where you can already see the philosophy of the model. Not one signal, not one metric, but multiple signals combined early. Their first build-out models connect all of this structural and sequence information to hydrophobic interaction chromatography retention times. This is basically a measure of how much the protein surface doesn't like water. And that turns out to be a handy signal. HIC retention time is a strong predictor of things like aggregation risk, solubility issues, and high concentration viscosity. All of the fun problems that tend to show up later and ruin your day. Their model fit their data very well and predicted HIC retention times better than any single feature model. Awesome! And once that worked, they asked the obvious next question. Does this generalize? So they tested a second parameter, high concentration viscosity. This measure is super essential for manufacturing and enabling subcutaneous injections. Imagine trying to pump or inject honey. Yuck. Tasty, but yuck. Wham bam, that worked as well. Now it looks like their approach is generalizable to multiple developability parameters. Suddenly, it's like the Regeneron scientists are at a shooting range with bottles lined up. Expression level, aggregation risk, formulation stability, bang, bang, bang. This is a framework that lets you test a huge number of potential antibodies, starting with just amino-acid sequences and some serious compute. Now here's the part of the paper that really won me over. The authors don't just tell you the model works. They tell you what it takes to run it. They include AWS pricing, about 8.5 hours and roughly $400 to process around 1,000 sequences, which is already pretty reasonable. But if that wasn't enough, they go a step further. They realize that full structure prediction is the slowest, most expensive part of the pipeline. So they ask another very practical question. What if we could predict those structure-derived features directly from the sequence? And that's precisely what they do. They train lightweight models, such as elastic net regressions, to approximate structural features without ever explicitly building the structures. And once you do that, the throughput explodes. Using this simplified version, they show the model running on a standard MacBook Pro, processing well over 100,000 antibody sequences. No cloud cluster, no GPU farm, just a laptop and time. At that point, this stops being a boutique AI tool and starts looking like something you could actually use to explore an entire antibody repertoire. This is the kind of thing a small startup could run, or an academic lab, or a single motivated scientist with a decent laptop and a long weekend. And that's why I love this paper. Not just because the work itself is solid, but because it paints a picture of what the future could look like. Wet lab researchers will still need to do the work to show that any particular molecule actually works. That part doesn't go away. But instead of running thousands of proteins through the lab to find 1 or 2 viable candidates, you might be screening a few dozen and still finding a winner. That shift alone could free up enormous amounts of time, money, and human effort. Time you can spend going after more diseases, money you can spend exploring better molecules, and effort that can be focused on making therapies that are genuinely better than what's currently in the clinic. Honestly, that's amazing. Positively wonderful. If you enjoyed this, I'd love it if you'd like the video, leave a comment or subscribe. It really does help the channel reach more people who care about this kind of science. And if you've worked with developability models, or you're thinking about using tools like this in your own work, I'd genuinely love to hear your thoughts. Thanks for spending a little time with me today. I'll see you next time.